Hi, everybody. I'm so pleased to be part of this very interesting session on heterogeneity within tumors that are labeled HER2 positive. Obviously, the treatment of HER2 positive breast cancer is one of the greatest success in clinical oncology, but we're still not getting to a cure for absolutely everybody. Hence, this issue of heterogeneity came up and led to the proposal to actually have an hour or so to dissect this issue out. So we're really pleased that we could be joined by three leaders in the field that will discuss different aspects of heterogeneity. The first talk will be in person by RM Blenman from Yale Cancer Center. And then Sarah Tolaney will join us virtually, followed by Aleix Pratt, who's here personally, but I think prefers to run his video, so he's word perfect. And then with a bit of luck, we'll have a little conversation at the end about some of the things that we've learned from your excellent presentations. So with that, I think I'd ask you to come up to the podium, Kim, and kick it off.
Oh, okay, so this is a little odd. Great. First and foremost, I'd like to thank the organizers for inviting me to give this talk. I'm a translational scientist. However, for this session, I've been asked to do a basic science talk. So what I will discuss today is a journey through the life of HER2. How do we get here, and where are we going? So here are my disclosures. So what we now call HER2 was first discovered in chickens in the 1970s. In the early 1980s, HER2 was identified as a glycoprotein with similarities to EGFR, and mammalian homologs were found. In the mid to late 1980s, it was discovered that HER2 was able to transform normal cells to cancer cells, and there were links to breast cancer. In the mid-1990s and early 2000s, biological functions were uncovered, and the complexity of HER2 signaling networks were presented. One of the biological functions uncovered during this period was HER2's role in normal mammary gland development. So HER2 plays an essential role in normal mammary gland development, and specifically in alveolar morphogenesis, which is the site of milk production and storage. Overexpression of the activated hybrid receptor containing the intracellular domain of HER2 can induce alveolar morphogenesis in the absence of growth factors. Transgenic mice expressing dominant negative HER2 failed to develop functional alveoli, while ductal branching was not affected. As a result of these discoveries, the early drug development focused on breast cancers that overexpress HER2, therefore are HER2-enriched cancers. HER2 positivity is a clinical definition, but there is molecular and clinical heterogeneity within the group. HER2 positive cancers can also be subdivided into different PAM50 subtypes that appear to have different clinical characteristics. Variability at the level of HER2 amplification appear to have clear clinical relevance. So Dr. Aleix Pratt will discuss this in detail as part of his translational talk in this session. HER2 is targeted in two fundamentally different ways. One, it can be a targeted agent that interrupts HER2 signaling. And two, it can be a targeted agent that is used as a target to deliver an antibody drug conjugate. So there is heterogeneity in therapy response. And Dr. Sarah Tolaney will discuss the details of some of these studies in her clinical session. Suffice it to say that PCR rates range between 17 to 77%, depending on ER status and treatment regimen. Complex multi-drug chemotherapy regimens have greater PCR rates than regimens with anti-HER2 therapy alone. So I will briefly touch on mechanisms of actions of monoclonal antibodies, tyrosine kinase inhibitors, and antibody-drug conjugates. First, I'll discuss just very briefly monoclonal antibodies. So trastuzumab is a monoclonal antibody that binds to the extracellular domain 4 of HER2. It disrupts ligand-dependent heterodimers or homodimers of HER2 receptors, which leads to a lack of intracellular signaling, which causes tumor cell arrest. It also induces antibody-dependent cell cytotoxicity by binding to the Fc gamma receptor 2 on cytotoxic immune cells, such as natural killer cells. In addition, it prevents HER2 shedding from plasma membrane of cancer cells. Pertuzumab is another monoclonal antibody that binds the HER2 extracellular domain 2. It acts mainly by inhibiting the ligand-mediated dimerization of HER2 receptors through steric hindrance. And then finally, margetuximab is a monoclonal antibody that binds HER2 and is engineered to bind the Fc receptor 16A, which is also known as Fc gamma receptor 3A, and not B, on cytotoxic immune cells such as NK cells. This produces a more potent antibody-dependent cell cytotoxicity stimulation and reaction. So other drugs and products are combined with trastuzumab to combat resistant mechanisms to trastuzumab. An example is CD44 hyaluronan polymer complexes were found to mask HER2 epitopes. Hyaluronidase degrades hyaluronan, and when combined with trastuzumab, allows trastuzumab to infiltrate tissue, enter the bloodstream, and bind to HER2. Pertuzumab was added to this regimen since it has a slightly different mechanism of action than trastuzumab. It also benefits from the ability to infiltrate the tissue, enter into the bloodstream, and bind to HER2. My colleague Sarah Tolani will discuss clinical outcome differences between these two trials in her talk in this session. So next I'll talk a little bit about tyrosine kinase inhibitors. So it is known that heterodimerization of HER2 with other HER family members provides mechanisms to escape inhibition of trastuzumab. Tyrosine kinase inhibitors prevent activation of receptor tyrosine kinases of the heterodimers, which prevents downstream signaling that would lead to abnormal cell growth and tumor formation. Lapatinib is a TKI that binds HER1 and HER2. Oops. Neratinib is a TKI that blocks HER1, HER2, and HER4. Tucatinib is a TKI that is selective for HER2, and it blocks only the HER2. So when we think about resistant mechanisms for HER2, there are markers that are sensitive and those that are resistant. Sensitivity markers that, if present, the chance of PCR is higher includes ER-negative status, HER2-enriched PAM50 molecular class, immune infiltration, HER2 expression levels. Resistant markers that, if present, the chance of PCR is low include ER-positive status, PI3 kinase mutations, PTEN loss, and HER2 copy number mutation, copy number a ratio less than 4.0, and focal HER2 positivity. In further efforts to address resistance, antibody-drug conjugates were developed. So antibody-drug conjugates, or ADC, couples trastuzumab with cytotoxic compounds through a chemical linker. This, in essence, provides a means to block the cells from growing via HER2, and also provides drugs that will help to kill the cells. TDM1 is an antibody-drug conjugate, or ADC, that contains a non-cleavable linker that attaches trastuzumab to a cytotoxic maytansine derivative that targets and disrupts microtubules. The Catherine trial showed that TDM1 was equally effective in HER2 high and HER2 low patients. This has potentially created a new breast cancer subtype called HER2 low, since this population of breast cancer cells is now targetable. Trastuzumab deruxtecan is an ADC that links trastuzumab to a topoisomerase inhibitor, which disrupts DNA replication. So, what are the new drugs in development? SYD985, which is an antibody, an ADC, links trastuzumab to a DNA alkylating agent that causes DNA damage in both dividing and non-dividing cancer cells, ultimately leading to cell death. Another compound, afatinib, is a TKI inhibitor that inhibits the HER1, HER2, and HER4, and certain EGFR mutants, including the exon 19 deletion mutations and the exon 21 mutations. So as we look at our milestones, we have gone through monoclonal antibodies, TKIs, and now ADCs to address HER2 overexpression and resistance. So what still needs to be considered? So what about heterogeneity in HER2 amplicon structure and mutations? So here I show some beautiful work from Dr. Matthew Ellis's group, highlighting the heterogeneity in amplicon structure through creation of proteogenomic maps comparing DNA versus RNA and DNA versus protein. They analyzed core needle biopsies from HER2-positive breast cancers before and 48 to 72 hours after initiating neoadjuvant trastuzumab-based chemotherapy. They identified potential causes of treatment resistance, including the absence of ERBB2 amplification and insufficient ERBB2 activity from therapeutic sensitivity despite ERBB2 amplification. So this leads us to the question of whether or not the heterogeneity in amplicon structure matter now that we have ADCs. And I think this is going to be a nice topic for our discussion as we wrap up the session. So in this same study from Dr. Matthew Ellis's group, proteogenomic analysis of HER2, positive tumors show that pseudo ERBB2 samples with ERBB2 amplification, but not protein expression. Some of these may have alternate 17q drivers, which is an interesting concept as we think about mutations. So where are we now? The current landscape of approved drugs include monoclonal antibodies, tyrosine kinase inhibitors, and antibody-drug conjugates. So where are we going? So new drugs in development include ADCs, TKIs, immune checkpoint inhibitors, vaccines, and biospecific antibodies, to name a few. So what have we learned? So development of effective anti-HER2 targeted therapies massively changed treatment strategies and prognosis of HER2-positive breast cancers. HER2-positive targeting is a testimony to precision medicine success. Anti-HER2 agents are now part of treatment in every setting of HER2-positive breast cancer. Tumor level heterogeneity impacts clinical pathology and treatment response. This includes HER2 in different breast cancer subtypes. It includes HER2 co-mutations with other oncogenes. And then it also includes HER2 amplicons, which are structurally highly variable, potentially as consequences of co-amplifications of other genes. More can be achieved. So ADCs, checkpoint inhibitors, vaccines, and bispecific antibodies may be the key to combating HER2 resistance. But we know that more can be achieved in the coming years to continue to improve outcomes and tolerability for patients with early-stage breast cancer and metastatic disease. Thank you. And I would like to acknowledge my colleagues here on stage, so Matthew Ellis, Sarah Delaney, Alex Pratt, and my mentors at Yale, Lajos Posai, David Rem, Miriam Lusberg, and David Stern, who was a pioneer in the HER2 biology and was responsible for some of the seminal work, for their assistance in helping me to prepare this presentation. Thank you.